But now it's my pleasure to introduce you to an individual with whom it's been a joy to work over the past few years. Let us please welcome Dr. Monique Pond.
Thank you for that warm welcome and introduction. Hello, everyone. Welcome to today's event. I'm Monique Pond, a team lead and program director at the National Cancer Institute SBIR Development Center. I lead some of our regulatory-focused initiatives that we have ongoing in collaboration with FDA, and in fact, it's one of those collaborations, our CARE program, that led to the creation of today's webinar. So I want to give a big thank you to Brenda Stoddard and SBIA. It's been wonderful working with you over the years and appreciate you hosting today's event, and to all my colleagues at FDA that have worked over the summer to make it possible today. I appreciate everyone's interest who's tuning in today. It really is exciting to see so many entrepreneurs dedicated to developing new technologies that can hopefully improve human health. So with that, for those joining in today that might be new to entrepreneurship, or maybe you're just new to SBIR, I'll give a very, very brief intro to the program and point you to where you can find more resources and opportunities for non-dilutive funding and additional information for small businesses. After this brief intro, then we'll get started with the main event. Thank you. The SBIR and STTR program provides R&D funding to small businesses that are trying to bridge the gap from basic science research to the commercialization of their products. And this is sometimes thought of as America's largest seed fund, with approximately $4 billion in non-dilutive funding that goes to support these small businesses each year. So at NIH, there's currently $1.3 billion in support available for small businesses. And of this, approximately $200 million of that's available through the NCI for companies that are focused on developing cancer-related technologies. To be eligible for SBIR STTR awards, a company needs to be organized as a for-profit small business that's located in the United States. If you're interested, you can find more details about eligibility that's on the Small Business Administration's website, and I've included a link here at the bottom of the slide to that. At the NCI SBIR Development Center, we oversee the administration of all the small business awards that are given at NCI, as well as provide non-funding resources to companies. So at any given time, we have about 450 to 500 active projects in our portfolio. And these companies are developing anything from a cancer therapeutic to an in vitro diagnostic, maybe therapeutic devices or a digital health technology. We also support agents related to cancer prevention as well. The vast majority of NCI-funded companies are developing products that will require either clearance or approval from FDA, so we really consider FDA as a valuable partner in the success of these technologies getting out to the clinic and hopefully helping patients. I often get asked about the makeup of our companies here at NCI. So I just want to touch on that a bit. Many companies have technologies that are in the preclinical stage, and the companies themselves were started within the last few years or sometimes even the last few months. So we do support, really truly startup companies. Often these companies could have fewer than 10 or 5 employees, sometimes even 1 or 2 employees if they're really new and starting out. And many NCI-funded companies are spin-outs of universities. So maybe you're out there, you're an academic researcher, and you are brand new to entrepreneurship. You are in the right place today. While these awards do go to the small business concern, funding can be used to support work that's done at the university through sub-awards. Now, we also have companies in our portfolio that enter the program when their technology is in the clinical stage. And some of these companies are also established small businesses that have been around for decades, maybe with quite a strong track record of entrepreneurship. So we have quite the spectrum of companies that we support. Now there's a lot of info in the slide I'm showing here. I just want to highlight that the current award sizes for NCI, we offer support ranging from $300,000 up to $4.5 million awards. And I've included the link at the bottom where you can find more details about how to apply for the specific funding opportunities. Lastly today, I just want to emphasize our cancer portfolio is really diverse. We think of ourselves as more technology agnostic, and instead we try to focus on the technology innovation itself and the potential unmet medical need it will solve. So if you're working on a cancer-related technology and you're interested in knowing more about NCI SBIR funding, definitely reach out to us. You can send us an email at ncisbir@mail.nih.gov. I think this is a great way to get started. Include a specific aims page or perhaps a short summary about your technology. You don't need to have already started your company to go ahead and reach out. We'll connect you with a program director and they'll meet with you and discuss your technology, try to answer any questions you have about the program. Now, if you're joining us today and you have a life science technology outside of cancer, we are very glad you're here. I've put a link at the bottom of the slide where you can find the list of program director contacts for all 24 NIH institutes and centers that have SBIR programs. So with that, I'd like to turn it back over to Brenda Stoddard, who will get our first session started today. It's focused on regulatory tips from the Center for Drug Evaluation and Research. Thank you.
Thank you so very much, Dr. Pond. And now I would like to provide a quick overview of CDER's Small Business and Industry Assistance Program, better known as SBIA, and our various resources. Learning objectives include locating the SBIA webpage and identifying 3 resources, and understanding how to register for SBIA events and find recordings of past events. Our mission revolves around you to ensure that you have immediate access to resources, education, and training, which allow for a more clearly informed and efficient developmental process with the goal of approving safe and effective human drugs and biopharmaceuticals. This slide here is just a visual representation of the global participation at SBIA events. Our resources include direct communication services, training resources, webpage, and news and events. Let's talk about direct communication services. Our staff comprises over 40 healthcare professionals, most of whom answer direct inquiries via the dedicated telephone numbers and the email address seen here, and these are our hours of operation. Last year alone, we responded to over 5,000 inquiries. We receive a wide variety of questions, and we respond very quickly. Whether it's providing you with a complete response, letting you know that we need to consult a different group within FDA, or guiding you on whom you should be contacting. On the phone, we'll even walk you through the website, help navigate you to where you need to be, assist you in identifying pertinent guidances, and troubleshoot various databases with you if at all possible. Let's talk about some of those training resources. We have trainings via workshops, conferences, and webinars such as this. In 2023, we hosted 8 conferences and workshops and 22 webinars reaching over 95,000 individuals representing 141 countries. 2 of our more popular annual conferences, and these two I have chosen because I think they are more pertinent to this audience, would be our Regulatory Education for Industry, or READY conference, which since 2021 has been offering 3 tracks: drugs, devices, and biologics. Then we also have our Clinical Investigators Training course, CITSI, which is not yet advertised but is tentatively scheduled for December 10th through the 12th. And I highly encourage you to subscribe to our listserv and register for the upcoming CITSI when registration becomes available. Another stream of information is our SBIA Chronicles. These are short 2-page electronic newsletters accompanied with audio podcasts which highlight different regulatory issues. Some past topics include Bayesian statistical approaches in the evaluation of drug products and considerations for drug products that contain nanomaterials. Next, let's go to our webpage. Here is a screenshot of our landing page, and this is the website here, fda.gov/CDER-SBIA. As you can see, it features our calendar of upcoming events with links to registration. Below the fold of the page are access points to our regulatory references, our LinkedIn sign-up, our YouTube library, and our SBIA Learn repository. And I would like to spend just a couple seconds on the SBIA Learn. SBIA Learn is essentially a database of training information for industry and could become a very good friend if you allow it. It currently curates 815 webcasts, which address a wide variety of topics, and you can link directly to the YouTube uploads from here. It is searchable by keyword and also by topic. The drop-down menu gives you a variety of different topics from biomarkers, chemistry, manufacturing and controls, clinical trials and research, and so much more. Perhaps the best resource relevant to this audience is our 2021 Oncology Therapy Development Workshop. And even though it's 2021, the information is still very applicable. Let's talk about news and updates. In addition to LinkedIn, we also disseminate information to industry via our email listserv or distribution list. We currently have over 160,000 subscribers, and we issue information daily regarding new regulations, FDA announcements and initiatives, webinars and conferences, many of which are SBIA hosted, but many are not. Here you see an ad for Ready 2024 previously mentioned, and these Ready conferences typically feature plenary with our 3 center directors. And also here to the right, you'll see a promotion for a recently established CDER Center for Clinical Trial Innovation, C3TI. And here is your action item for this presentation. On our main page, again, fda.gov/CDER-SBIA, you can find a link to sign up for the emails and do so even now. If you scroll all the way to the bottom, here you see our contact information again, but here you can enter your email address even as I'm speaking. Enter the email address, hit subscribe, and you'll be getting emails from us as early as tomorrow. Here are the QR codes for the various resources previously mentioned. Here is a summary and call to action of all the contact information and the ways to follow us and subscribe to our listserv. I want to once again introduce our moderator for today, Lieutenant Commander Nora Lim. And I would also like to introduce our upcoming 4 CDER presenters. We have Dr. Parisma Patel from the Office of Pharmaceutical Quality, Dr. Emily Warren from the Office of New Drugs, Dr. Caitlin Tidens from the Office of New Drugs. And first up with his presentation, An Oncology Drug Development Mindset, First Steps, is the inimitable Dr. Jeff Summers, Associate Office Director of the Office of Oncologic Diseases within the Office of New Drugs. Please, let us welcome Dr. Summers.
Thanks, Brenda. Dr. Stoddard. As you'll hear, I can't say enough good things about the efforts and programs you guys at SBIA and your counterparts in SBIR and CDER provide. Good morning, everyone. My name is Jeff, and I'm the Associate Director for Translational Sciences in the Office of Oncologic Diseases in CDER. I'm excited that SBIA is producing this webinar and for the participation of SBIR, CDER, and CDER, who have all been part of the NCI CARES program. I personally have had the opportunity to interact with you, the academic investigator, life science accelerators, and small oncology startup companies, and I know that real pharmaceutical innovation, at least half the time, originates with you. The pathway, the target, the epiphanies frequently are your ideas, your experiments, your discoveries. I have read and heard a lot of your questions through various OCE programs and scientific conferences. I'm going to try and distill successful, effective drug development down into one primary bullet, 2 take-home points, and a bonus imploration. That will no doubt be a recurring theme today. What I hope to accomplish in 10 minutes with a minimum of metaphors is to put a positive spin on the questionable webinar title, to telegraph in no uncertain fashion the action you need to take to be successful with a caveat, to highlight the approaches and opportunities to accomplish that, and even though it's probably not AI, another caveat, and finally, a bonus imploration. I'm assuming somewhere in the development of this webinar that the title went through or by me and I did not say anything. Now, the communications folks in the Oncology Center of Excellence are always telling me, you cannot be negative, Jeff, just say the positive. And I always argue that this is not reality and instead just state facts, data, truth in a pleasant manner. But as I perseverated over that title, I realized these are not really your do's and don'ts, but the agency's FDA's limitations and mandates. We, FDA Oncology, are required to follow statutes and regulations. And I am not going to quote you a bunch of excerpts from Section 300 of the Code of Federal Regulations. But I will highlight and perseverate on the guidance documents we generate to help you follow regulations. But let me start with a positive. The Oncology Center of Excellence wants nothing more than to help you add more vibrant and effective colors to the palette of anti-cancer therapies. That's our mission, our primary due, to approve more safe and effective drugs. And unlike the approach of the past, which we now know was hopeful but misguided, where the idea of more is better was incorrect, and we have a whole program called Project Optimus to move away from the maximally tolerated drug paradigm of the past. More is actually better. More drugs targeted to specific cancer cell molecular vulnerabilities increases the possible log cell kill and heterogeneous tumors, getting us closer to cures. The Oncology Center of Excellence's primary due is to approve safe and effective oncology drugs. It's also required from a regulatory perspective that we, FDA Oncology, make it a fair and level playing field. Your company doesn't receive any help that large pharma cannot also avail themselves of. They just don't need the regulatory help. They have in-house experts who have been around the block hundreds of times. And finally, we would like to guide you in meeting the standards of manufacturing a safe, pure, potent drug that performs as purported in your proposed product label. So I have seen many of your questions, and FDA and the OCE really want to help. We do not want to be a scary black box, but there are things we can't do, answer, or provide. We don't do discovery. This originates from your creativity and insights. The rush of eureka is yours alone. We are not consultants. In general, we will not tell you what path or approach to take in obtaining what we think is necessary data to support the safety and efficacy of your drug. There are multiple ways to skin a cat. We will not disclose information from other entities' drug development programs, period. If there is a safety issue with a particular product type, pathway perturbation, mechanism of action that we think will be relevant to your drug, we may ask for specific safety data to address those concerns that arose from other drug development programs. And monetary matters are outside of the FDA Product Center's purview and expertise, and intellectual property due diligence is your responsibility. The one action for success, the number one action to take is, but first the caveat. Now this is a little simplistic, but there's an adage for successful drug development, and it starts with one that works. So I can give you a whole talk on what I think that magic sauce is, but what is just as important is you, your team. And the number one action is, I hope this comes across in a collegial and respectful fashion. You need to step up to the plate and inform yourselves. As you all know, there are limited resources, particularly in regulatory agencies. We, FDA Oncology, can only do so much based on those limiting resources. You've done the creative part, the difficult part. Science is hard. You've identified a pathway, an epitope, a target. Pick your arrow, whatever approach you're going to use to hit that target, and turn it into a drug. You are in the major leagues now. Take home point number one. Read the relevant FDA and ICH guidance documents. All kinds of people spend all kinds of time writing these documents so you can understand what needs to be done to meet the regulatory standards. These guys at all, the Oncology Center of Excellence and cross-center offices, have created 60 new oncology-specific guidance documents over the last 6 years, all linked on the Oncology Center of Excellence's guidance webpage. Whenever I give one of these entrepreneurial talks, I bring this slide up. Each one of these groups is here today, and they produce invaluable educational resources that they will no doubt discuss today. From SBIR's OTP Learn, which I am summarily impressed with, to CDER webinars like this one today. It's out there. You just need to Google, listen, watch, and learn. One of the many educational endeavors the Oncology Center of Excellence sponsors are the Project Catalyst Bench-to-Bedside Chats, designed specifically for academicians and university life science accelerators, where we discuss topics related to early-stage drug development. And not to be FDA-centric, both NCI SBIR folks and the NIH Seed Office have produced fantastic educational resources for the life science entrepreneurs for CDER, SBIR, and CDRH regulated products. Take-home point number 2. I'm assuming you, the audience, are creative scientists or physician scientists who have a day job and then some, and that you have made interesting observations in your science and want to exploit those observations against cancer and translate the science into medicine. And hopefully you have done the Crux activity experiment. You have formed a company to make a product to treat cancer. You are an oncology startup. Congratulations. Many times, startup companies fail long before their science fails because of poor enterprise. And navigating the regulatory aspects of drug development is one part of that enterprise. And take-home point number 2 is, most founders of biotech startups realize this quickly. They have a day job and they like their day job. Maybe you could do it all in your academic lab in the rented wet space you just leased last week. Maybe you and the smattering of actual employees of said startup could learn on the fly. But you are likely to make multiple mistakes in navigating the enterprise of drug development. If you do not have the expertise in-house, take-home point number 2, carefully consider bringing a competent regulatory consultant to the table. And finally, my bonus imploration. You may have conducted seminal science and generated a potential novel therapeutic, but sometimes we notice that you might lack experience in the specific disease indication you are pursuing. Engage with colleagues, particularly your oncology subspecialist and first-in-human clinical trialists. And engage with FDA. You are the pharmaceutical innovators. It doesn't happen without your creativity. But we, FDA Oncology, and the OCE and the projects and programs they're in, and most importantly, the people, want to help you advance oncology patient care. We are not a roadblock, or box checkers, or bureaucrats. We are scientists, oncologists, nurses, pharmacists, and most importantly, all of us are cancer patient advocates. There are more different ways to engage with FDA Oncology today than there have ever been. We are not a black box or something to be feared. The Oncology Center of Excellence, through Project Catalyst, conduct informal accelerator innovator discussion aid meetings with academic project teams and coordinate the oncology regulatory expertise and early guidance, or OREG program, to address product type development questions you might have. We, FDA Oncology, also conduct slightly more formal pre-IND meetings, also known as Interact meetings, to help you with early stage drug development issues. And there is a pre-IND meeting to make sure you are generating the required IND enabling data and not conducting unnecessary studies. I don't think there are any PDUFA fees associated with these programs. They are a free lunch, a free resource. So a quick summary. Step up to the plate. The fact that you are here today suggests you are at least in the on-deck circle. Read, listen, learn. Consider competent consultation sooner rather than later. And most importantly, engage, or you might find yourself stuck in space. And I leave you with my favorite quote: research is to see what everybody else has seen and to think what nobody else has thought. Albert St. Georgi. I implore you to think differently in your research and to listen carefully in your drug development efforts. And with that, I'll turn the virtual floor over to Dr. Parisma Patel, who has been an instrumental and insightful subject matter expert for your product quality questions. Dr. Patel.
Good morning, and thank you for joining me. My name is Dr. Parisma Patel, and I'm a Division Director in CDER's Office of Pharmaceutical Quality. Today, I'll be giving an agency perspective on chemistry, manufacturing, and controls, regulatory considerations, and resources. Today, I'll start off with a brief definition of pharmaceutical quality. I'll discuss how chemistry, manufacturing, and controls, or CMC, changes through clinical development. I'll provide some key regulatory definitions. I'll touch on some specific considerations for drug substances and drug products. And I'll end with important guidance documents and resources. I'd like to start with an overview of the mission and vision of CDER's Office of Pharmaceutical Quality. Everyone deserves confidence in their next dose of medicine. Pharmaceutical quality assures the availability, safety, and efficacy of every dose. One challenge to the manufacturing aspects of an application is that it covers the entire life cycle of the product and is often rapidly changing from an IND stage of development to commercialization. During preclinical stages of development to the point of IND submission, there is typically limited CMC information. As a product's development proceeds through the phases of clinical development to pivotal clinical studies, significant CMC development is happening. CMC development has to be at a stage to assure consistent manufacturability to support a marketing application. Following approval, the CMC information continues to change as suppliers or manufacturers may change, and the manufacturing processes may continue to be optimized. Throughout this process, the CMC team is meeting with sponsors to advise on key aspects of CMC development as they move closer to commercialization. This includes early-stage interactions such as Interact and pre-IND meetings. The CMC team is involved for Type B meetings after each phase of development, and CMC-focused meetings can happen throughout clinical development. Most CMC-focused meetings typically happen around the end of Phase 2. I'll provide a couple of key regulatory definitions that can be found in the Code of Federal Regulations. Shown here is the definition of drug substance, and to summarize, the drug substance is the active ingredient that provides the pharmacological activity or direct effect for the diagnosis, cure, mitigation, treatment, or prevention of a disease. This is essentially the active component of the drug product that affects the human body. The drug product is the finished dosage form, and it contains the drug substance, and it may or may not contain other ingredients. CMC information required for an IND submission are outlined in the Code of Federal Regulations. The main text of the CFR can be broken down into a few requirements for CMC. They include description, composition, and controls of the drug substance and drug product, information on the manufacturer, the manufacturing process, and data to support stability, information to assure identity, quality, purity, and strength of the investigational drug. The regulations specifically emphasize the identification and controls of the new drug substance and raw materials for an initial Phase 1 submission. Guidance documents are routinely published on FDA's websites. These documents provide FDA's current thinking on a topic and outline information that should be provided in IND and NDA submissions. Guidance documents clarify the type, extent, and reporting of CMC information to ensure quality of the proposed clinical studies. These documents help to outline the data needed for regulatory development and are only recommendations. If sponsors veer away from this advice, they should provide justification and scientific reasoning and meet with FDA if needed. I'll start off focusing on the drug substance or active pharmaceutical ingredient information that FDA generally expects as part of an IND application. First is general information. This contains information about the structure, molecular weight, formula, and the source of the drug substance. The manufacturing process section includes a description of the chemistry for how a drug substance is made and includes both a flow diagram and a written description for each step of the manufacturing process. The characterization data section includes both structural characterization data and characterization of physical-chemical properties. The impurities section provides available information on observed and potential impurities. Sponsors should specifically address differences in impurities between toxicology and proposed clinical batches. The controls part of CMC defines a release specification, which is a predetermined set of criteria used to release batches of drug substance for use. This section includes information on the batches intended for use in the clinical study and those that were used in the toxicology study. And finally, data to support stability for a drug substance should be provided in the stability section. Preliminary stability data is typically sufficient for an initial IND submission. I would like to now touch on specific information that is needed for a drug product compared to a drug substance as part of an IND submission. For the drug product, it's important that we have a description of the dosage form and any information to justify the safety of a novel technology or any complex drug product formulation. It's particularly important to clarify any patient administration information for the proposed dosage form. For example, a drug product that is administered parenterally, it'd be important to consider compatibility issues with any administration devices or solutions that may be used to dilute the drug product prior to administration. The quantitative composition of the drug product should be provided, and this includes a list of the inactive ingredients or excipients. We ask for information on the compendial status or quality of those excipients. If novel excipients are used, additional CMC information may be needed to assure quality of those excipients. Additional information may also be needed to assure safety of any animal-derived excipients. If using novel excipients, we generally recommend discussing your approach during a pre-IND meeting to assure adequate data is provided in the IND submission. The drug product manufacturing process should be described with both a brief written description and a flow diagram. And additional information on the sterilization process should be provided if it's applicable to your drug product. Similar to the drug substance, the control of drug product is essentially a specification that includes a set of criteria to demonstrate quality of the drug product prior to releasing batches. This section includes information on impurities that are specific to the drug product, as well as the proposed batch data for use in the intended clinical study. Finally, information on how the drug product is packaged and preliminary stability data should be provided in the IND submission. Some of the CMC issues that are likely to lead to clinical holds of ongoing studies include the following. Questions related to adulteration or purity of the materials used to manufacture the drug substance or the drug product. If the impurity profiles are not sufficiently characterized or the safety of the impurities is not justified by supportive non-clinical studies. Any impurities that are known to be highly toxic or pose potential risks to patients. Unreliable analytical methods that may undermine the confidence in the data provided in the IND. Insufficient batch data to demonstrate manufacturability of either the drug substance or drug product. Any significant stability issues. Lack of sterility assurance or endotoxin controls could pose significant safety issues or risks to patients. Challenges with the drug product formulation, such as formation of particulate matter upon storage of a parenteral drug. And finally, issues related to cGMPs at the manufacturing facilities. One way to avoid issues at filing of an IND includes consistent communication with the FDA through industry meetings and paying close attention to the advice given during those meetings. Prior to an IND submission, every sponsor is granted at least one pre-IND meeting. As part of this meeting, the sponsor of the IND submits a brief package that describes the background of the product and proposed clinical development. CMC-specific focus areas during a pre-IND meeting include the following: whether the manufacturing process and characterization data appear reasonable, the acceptability of the drug substance and drug product controls or specifications, the overall stability data and stability design, whether the impurity levels and controls appear reasonable for a phase 1 study, adequacy of the toxicology batches to support use of the proposed clinical batches, and any overall potential gaps or hold issues that the CMC team may find. I would like to summarize with some regulatory do's and don'ts. Do read and apply available guidance documents. Meet and engage with FDA early, especially for complex drug substances and drug products where there may be little applicable guidance documents available. Ask focused questions relevant to specific regulatory challenges during meetings. Address FDA advice and comments that are provided during development. Engage the CMC team early. Assure quality suppliers of raw materials, excipients, and other aspects of your product, and pay close attention to the cGMP requirements. FDA spends significant time providing comments during IND review and during meetings. Don't ignore FDA advice and input. I would like to end with a few resources, including important websites as resources, IND-focused guidance documents with CMC advice, and resources for expedited programs. This slide provides resources that are specific to control strategies for impurities that could be used during development. With that, I would like to end my section today, and I will pass it on to Dr. Emily Warren, who will be providing a non-clinical overview.
Thank you, Dr. Patel, for that introduction. I'm Dr. Emily Warren, a pharmacologist and acting non-clinical team leader in the Division of Hematology, Oncology, Toxicology, or DHOT, at the FDA. Today I'll be giving an overview of CDER non-clinical resources and relevant guidances that are key to approaching first-in-human studies in oncology. Here's an overview of today's webinar. We'll discuss where to start, what we are looking for at the FDA, any potential pitfalls you may encounter, and general recommendations for your consideration. In general, non-clinical data includes pharmacology or mechanism of action data, pharmacokinetic data, and toxicology data. The role of a non-clinical reviewer at the FDA is typically front-loaded, such that we review non-clinical data submitted under new INDs to determine an initial safe starting dose for a first-in-human clinical trial, as well as what toxicities to monitor for in the clinic. Importantly, we function as part of a multidisciplinary team. So where to start? You've identified a promising target and shown that your drug affects that target. You've shown activity in an in vivo tumor model. But is that enough? And what's next? Well, we recommend that you start with the guidances. This slide contains titles and links for the non-clinical guidances that are most relevant to oncology development. Specifically in oncology, we use the ICHS-9 and the ICHS-9 question and answer guidances, which discuss non-clinical evaluation for anti-cancer pharmaceuticals, and this includes small molecules and biologics. Biologics are also discussed in ICHS-6. The ICHS-9 guidance explains the basic recommendations for anti-cancer drugs. These include 28-day GLP-compliant toxicology studies in 2 species, or a single study for biologics if you have scientifically justified that there is only one pharmacologically relevant species. These studies should use the intended clinical route of administration, and the dosing frequency should match or exceed that intended for clinical administration. However, deviations can be justified. In general, these GLP-compliant toxicology studies are the primary data used to determine the starting dose for first-in-human trials, although occasionally a general toxicology study is not conducted if there is no pharmacologically relevant species. And a useful tip here is that you can initially submit draft toxicology reports containing signed histopathology reports to the IND, and the final quality assured study reports would then be expected within 120 days of our receipt of the IND. ICHS-9 includes an exception on standard toxicology-based methods for determining the starting dose of immune agonists. It states, for biopharmaceuticals with immune agonist properties, selection of the start dose using a minimally anticipated biologic effect level, or MABEL, should be considered. A MABEL approach relies heavily on pharmacology studies. And in general, it's important to use the totality of data to justify the proposed first-in-human dose. DHOT has numerous publications evaluating methods of first-in-human starting dose selection for different types of products, and these have resulted from internal research projects. These publications are shown on this slide for your reference. In terms of toxicology study design, refer to the CIFSAN Red Book for detailed descriptions of toxicological endpoints for a large array of study types. So key endpoints in GLP toxicology studies are shown on this slide. and additional endpoints can be included if warranted based on your drug's mechanism of action. So, for example, cytokines could be measured as needed for immune-activating products. Good laboratory practice, or GLP, is a key concept in non-clinical development. GLP doesn't just refer to a study with good controls and endpoints. It also includes details about archiving, study conduct, and responsibility. GLP is described in 21 CFR Part 58 in the Code of Federal Regulations. Prior to initiating GLP studies, consider the contract research organization or lab that you want to conduct your studies, and make sure they can format the data in SEND, which is now required by the FDA for multiple study types. In addition, we encourage you to plan ahead. These GLP studies can be expensive, but they really are foundational for drug development. If you have questions or uncertainties about GLP study design, consult an expert first. You can also request a pre-IND meeting with the FDA before initiating the study. We can provide general feedback on major red flags and missing endpoints, but we do not design the study for you. So what else is recommended for a first-in-human study? Well, it depends on your product. According to the 1995 Content and Format of INDs guidance, INDs should contain proof of concept data, including a description of the pharmacologic effects and mechanism of action of the drug if known. We also recommend providing screening data and looking for primary and secondary targets, concentration response data, and in vitro cytokine release data for immune agonists. Safety pharmacology data, including CNS, respiratory, and cardiovascular assessments, are also recommended, and in vivo safety pharmacology endpoints are generally built into toxicology studies for oncology drugs. I'll now discuss some stumbling blocks and pitfalls that sponsors may encounter when submitting a first-in-human IND. According to a 2020 FDA publication, the most common non-clinical reasons for putting a new commercial IND on hold in oncology are concerns with the first-in-human starting dose and/or dose escalation, missing studies or supporting information, and severe toxicity in toxicology studies. Speaking of starting doses, we expect sponsors to scale by body surface area for small molecules and antibody drug conjugates, and by body weight for large biologics. If the drug is an immune activator, we expect pharmacology data to be submitted to support the selection of an appropriate starting dose. Potential pitfalls can also be GLP related. For example, your GLP toxicology study reports need to contain a signed pathology report. Only signed pathology reports are considered raw data. In addition, it's important to provide data supporting your selection of pharmacologically relevant species for toxicology studies that are conducted with biologics and oligonucleotides. And again, if needed, don't hesitate to consult an expert. So for your reference, this slide describes special considerations and additional resources, including guidances specific to radiopharmaceuticals, novel excipients, and botanicals, as well as information pertaining to antibody drug conjugates. And finally, we encourage you to request a pre-IND meeting. We now have Interact meetings, which can be requested to get FDA input on unique challenges or the use of novel alternative methods, or NAMs. A traditional Type B pre-IND meeting can also be requested, with follow-up opportunities available, including written clarifying questions. We recommend that your meeting package includes enough data for us to answer your questions, but don't submit full study reports, as these are generally not reviewed at the pre-IND stage. In addition, although we will provide feedback on your proposed approach to determining the first-in-human starting dose, We will not agree to a specific starting dose at the pre-IND stage because we have not yet reviewed the full study reports. So in summary, there's a lot of non-clinical data needed to open an IND. Let ICHS-9 be your starting point. Invest in good GLP-compliant toxicology studies. Seek out a regulatory consultant if needed. And request a pre-IND meeting. As a closing thought, we hope you use these tips to improve your non-clinical package for your next first-in-human IND submission for oncology indications. This concludes my presentation. I'll be back shortly to answer your questions. Now I'd like to pass it on to Dr. Caitlin Tidings.
Thank you, Dr. Warren. As mentioned, my name is Caitlin Tidings, and I'm a pediatric oncologist and medical officer within the sarcoma team in the Division of Oncology 3. Today, I'll be providing general clinical considerations for first-in-human trials. We will start briefly going over some general good practices, followed by discussing important components of new INDs, common mistakes we see when reviewing new INDs, and we will finish with resources you may find helpful. Here are a few good practices that one should be aware of when developing a first-in-human trial. It is very important to engage a trialist or trialists who have expertise, and this is ideally in both the disease indication as well as with first-in-human trials. Next, pre-INDs can be requested to discuss if you have enough of the package to submit an IND, and this may help streamline the IND review process. Throughout the development program, it will be important to distinguish drug activity, which reflects the biologic effect of the drug, from clinical benefit, which reflects clinical effect that is meaningful for a patient, and failure to do so may waste resources. With that in mind, we will jump into an overview of INDs. As many of you know, there are three major components of the IND: chemistry, manufacturing, and controls, or CMC, preclinical data and analysis, as well as the protocol itself. The review team at FDA is multidisciplinary. Regulatory project managers facilitate all communications between the sponsor and FDA, as well as between the FDA reviewers themselves. Our chemists review the CMC portion, toxicologists review the preclinical data, and pharmacologists, clinicians, and statisticians review the protocol. FDA teams discuss new INDs at weekly safety meetings, and we send information requests or IRs to the sponsor for revisions or clarifications. These can include potential hold comments, which are changes that must be made by the sponsor and pertain to safety, or they could be non-potential hold comments, which are suggestions by the FDA that the sponsor should think about. Following these communications, the study may be placed on clinical hold, or the study will be declared safe to proceed. For first-in-human and exploratory or proof-of-concept trials, FDA focuses the review on safety. If we determine a new IND to be safe to proceed, it may be with or without contingencies, and these are usually discussed during the back and forth of IRs. For example, the trial may be contingent on uploading a revised informed consent document. If we determine a new IND is not safe to proceed, the IND is placed on hold. Partial holds include allowing the trial to proceed up to a certain part. So for example, up to a certain dose level because there was insufficient safety data to allow escalation beyond that. After which the sponsor must discuss with FDA and address FDA's concerns before continuing. Complete holds require that sponsors address all potential hold issues before the trial can begin. These are the various components of the IND we will briefly go over. For dose escalation, one can follow a rule-based design such as a 3+3 and its variants, accelerated titration design, etc. These are often easier to implement. For example, 3+3 is very commonly used, but they may not be the best at determining the maximum tolerated dose and the recommended phase 2 dose, and more patients may be treated at a subtherapeutic dose. Model-based designs use statistical models, including continuous reassessment model, CRM, modified CRM, Bayesian optimal design, etc. These are better at determining the MTD and RP2D, but are more challenging to implement and require biostatistical support for decision making. Dose escalation may be more rapid, which risks them being too aggressive. And if there's no overdose control, you may treat more patients at a toxic dose. It is important to talk to your statistical colleagues about the best design to use and what your institutions and programs can accommodate. The goal of dose escalation is determining the RP2D, the MTD, or if the MTD is not achieved, the maximum administered dose, or MAD. The RP2D is the dosing regimen that you decide should be further studied based on clinical PK and PD, safety and tolerability of the dose, and preliminary efficacy if you looked at that. One thing to consider is if the drug is being taken orally or is going to be taken daily or frequently, long-term tolerance and toxicity should also be taken into account. If you're using the 3+3 design, the MTD is defined as the dose at which 0 out of 3 patients experience a dose-limiting toxicity, or 0 to 1 out of 6 patients experience a toxicity. Other designs have other definitions. Generally, we want the highest probability of risk of drug toxicity to be less than 30%, and the target risk of drug toxicity should be aimed at about 25%. But there is some flexibility depending on the design, so we will provide feedback if needed during our review of the protocol. Acceleration of development beyond dose escalation can take traditional approaches such as activity estimation designs. These can be single-armed or randomized. For single-arm, use a design that would limit exposure of a large number of patients to ineffective drugs such as a Simon 2-stage. And for randomized designs, use a common control arm and avoid formal comparison between experimental drugs. Some newer strategies include master protocols, including basket trials, which looks at multiple cancer types with a common genetic mutation like NTRK, or umbrella trials, which looks at a single cancer type but evaluates multiple drugs. You can refer to the guidance at the bottom of the slide regarding enrolling patients for clinical trials investigating drugs in the non-curative setting and what you can include in the informed consent document regarding available therapy outside the trial. We have done a lot of work to expand eligibility criteria and allow patients the option to receive an experimental drug. The focus has been on later phase trials, but even first-in-human trials could benefit from careful consideration of eligibility criteria to improve patient access to trials. For dose selection, the dose range is guided by non-clinical toxicology, and dose escalation can be linear or logarithmic. Generally, linear increases are used for small molecules with up to 100% increases, and logarithmic increases are used for proteins. If we expect a fair amount of toxicity, we will recommend conservative dose increases. FDA is working hard to find ways to better optimize dose selection, and we have a working group called Project Optimus undertaking this very important task. Dose-limiting toxicities should be included in the protocol by way of defining the observation period, which should be at least 1 month, defining what a DLT evaluable patient is, with some example language here, and listing the DLT criteria itself. This is where it can also be helpful to have a pre-IND meeting, as there's often some back and forth regarding DLT criteria. Dose modifications must be included and based on anticipated toxicities and/or the dose-limiting toxicity. You can also consider dose reducing to the previous tested levels in the trial. Just a quick note for safety monitoring, you must monitor and collect all adverse events during first-in-human trials. This is for up to 30 days from the last dose if a drug is not immunotherapeutic, and up to 90 days from the last dose if the drug is an immunotherapeutic, and if delayed onset of adverse events is possible. The parameters to monitor should be based on what's known about the drug, the anticipated toxicities, and the mechanism of action, and should be a minimum of a CBC and CMP and more specific testing based on what I mentioned. Both subject stopping and trial stopping criteria must be included in the protocol. I will quickly note that terminating the trial for safety is usually a decision point that is already part of the dose escalation, but bear in mind that the MTD may not be reached depending on the toxicity of the drug. Finally, I put a short list of some common pitfalls I see when reviewing INDs. This is not exhaustive. For example, some protocols we see state that they collect AEs related to the study drug. However, all AEs should be collected regardless of relatedness, as all AEs are considered related to the study drug in first-in-human studies. We often make notes of aggressive dose escalation and not starting at least dose -1 for combination regimens. I've included some helpful resources that you can refer back to for FDA websites, guidances, and where to find templates. And I want to thank you all for your time today. We will next be moving to the Q&A portion of this session. Thank you.
Thank you again to our presenters for their excellent presentations and insights. We will now begin our question and answer panel. If you haven't had a chance to enter your question into the Q&A chat pod, please do so now. We'll answer as many questions as time allows. And now we'll go on to our first question. That looks like it came in for Dr. Jeff Summers. Dr. Summers, is there an equivalent office or program to the oncology regulatory expertise and early guidance for other therapeutic areas?
Thanks, Nora, for the question. The rare disease program at FDA has a lot of programs aimed towards trying to help the development of drugs for rare diseases, because they're a lot of times economically infeasible to develop. And so I don't know specifically what those programs are, but I know that they're very interactive, and I can't highlight more the positive impact that I think that they have also through the Office of Translational Sciences but we, Oncology developed the OREG program through the Oncology Center of Excellence with, Rick Pasder's help, and I'm not aware of any similar program across the other OND divisions, except for the rare diseases program. But there are a lot of different programs, and I may be unaware of everything.
Right. Thank you so much for that response. We have another question for you, Dr. Summers. Is it mandatory to have a pre-IND meeting before the initial IND submission?
Hey, thanks, Nora, for that question. Yeah, no, I saw that. There's a couple other ones I might touch on too as well, but absolutely not. I mean, you can come in with an IND. I have sat on a number of panels, and one of them was at AACR a few years ago, and a very, very competent CEO of a biotech oncology drug company was it came along with the idea was they had to hurry. Their drug was, they were in competition with everyone, and so they did that, and they got put on hold, and he was, and then fortunately those are kind of things that need to be reported to various agencies outside of FDA in the United States, and that may not necessarily look great for your development program. So I would highly recommend that you take advantage of every chance to interact with the FDA, whether that be aid meetings, the OREG program, the pre-IND and the pre-IND meeting during your development plan, so that you can be as informed as possible and understand what those roadblocks are, and so that you can go around them and navigate them more easily. Thanks.
Thank you so much for that information as well. And now we'll address some questions that came in for Dr. Pareshma Patel. Dr. Patel, during the development timeline for small molecule therapeutics, at what stage in the interaction with the FDA do we reach agreement about which steps in the synthesis should be GMP?
Thank you for this question. It's probably one of the top questions that we get from the CMC discipline. So I want to start by just pointing out the regulations that actually govern current good manufacturing practices, or CGMPs. Those regulations are outlined in 21 CFR 210 and 211. And these regulations are really focused on commercial manufacturing and and may not be appropriate for phase 1 studies. So for phase 1 studies, in my slides, I did include a link for the phase 1 investigational drugs CGMP guidance, and that provides some of the requirements for phase 1 studies and CGMP requirements for phase 1 studies. Now, when it comes to understanding how much of your manufacturing process should actually be conducted under CGMP, what we recommend is that sponsors come in and have a CMC-focused meeting to discuss their manufacturing process to support pivotal clinical studies and ultimately commercialization. And we recommend this prior to those pivotal clinical studies being run. So usually the end of phase 2 is when that happens. And the FDA assesses the CGMP process based on the guidance ICHQ 11. And there's also a associated questions and answers document as part of that guidance as well. And that helps to determine the appropriate starting materials for a CGMP process. So I want to actually just end by kind of reiterating that usually for phase 1 studies, we do not expect that the manufacturing process at that stage is going to be your commercial process. And typically we see a minimal number of steps under CGMP for phase 1 or the first in human study. And as development nears commercialization, we see additional steps are added or needed in order to assure that the manufacturing process meets the ICHQ 11 requirements. And with that, I want to thank you for that question.
Thank you so much for that response, Dr. Patel. We have another question that came in for you as well. Is a COA mandatory for the initial IND clearance?
Thank you for that question. This is a great question as well. For an initial IND submission, we generally need to see some demonstration that the drug substance and drug product can be manufactured in order to support the proposed clinical study. So the actual certificate of analysis for the batches is suggested, but it's not necessary. A summary of the batch data associated with those batches intended for the clinical studies, as well as those that were used in toxicology studies, should be submitted in the IND preferably in a tabular manner, clarifying the intended use of the batches as part of the IND submission. However, the actual primary COAs don't necessarily have to be submitted. Thanks.
Thank you so much for that information as well. Now we'll address a question that came in for Dr. Warren. Dr. Warren, can you say more about how you recommend drug developers use the Red Book?
Thank you for that question. So you can access the Red Book online, and there you'll find descriptions of numerous types of toxicity studies, including general toxicology studies, genotoxicity studies, and reproductive studies. And the Red Book describes detailed methods for conducting each of these studies, and so you can use that to help you design your toxicity studies. Thank you.
Thank you so much for that response. We have another question for you, Dr. Warren. For FIH dose selection for oncology and immuno-oncology, can the NOAEL approach also be used? Okay. Sorry, Dr. Warren, if you're speaking,
I apologize.
No problem.
Sorry. Thank you for that question. No, we generally do not use the no observed adverse effect level from toxicology studies to determine the first in human starting dose for oncology for immune activating products. So for immune activating products, we use a MABEL approach, which is often very centered on in vitro pharmacology data using human cells. And our approach also includes the totality of the non-clinical data in your submission. Thank you.
Thank you so much for that information as well. And now we'll address a question that came in for Dr. Caitlin Tidings. Dr. Tidings, while not mandatory, how often do you request the ICF in an initial IND submission?
Thank you for the question. That is correct. It is not mandatory. It must be stated within the IND submission how informed consent will be obtained, but the document itself is not required. However, I will say that the majority of INDs we do review have informed consent documents included. And depending on the review of the safety as we go through the IND, we may request it if it was not already provided. But I would say the majority already come with them. Thank you.
Thank you so much for that response as well. And now we'll go back to Dr. Patel for another question. Dr. Patel, is it still, sorry, how many months of stability data for a drug substance needs to be included into the IND application?
Thank you. This is a great question. So I want to first reference our phase 1 CMC guidance, or what I call our phase 1 CMC guidance. This is essentially the guidance named content and format of INDs for phase 1 studies of drugs, including well-characterized therapeutic biotechnology derived products. Within that guidance, there is the CMC section that really outlines the requirements for phase 1. And within the section for stability, it does outline that typically for a phase 1 study, what we need is preliminary data based on representative material for both drug substance and drug product. And so there's no strict requirement for the number of months of data that's necessary for a phase 1 study, but the guidance does outline that we need preliminary data on representative material for both of those, for both drug substance and drug product. So I think that as long as there's sufficient data to support the proposed clinical study, to support stability, and then we would need to have stability updates as the clinical study is ongoing. So it's really up to the sponsor to assure that the material going into patients is going to meet the specification at the time that it's dosed. Thank you.
Thanks so much for your response, Dr. Patel. Now we'll have a question for Dr. Jeff Summers. Dr. Summers, how would you interact with FDA if you ran a startup oncology company?
Thanks for the question, Nora. That would be great. Yeah, I don't think that's going to happen. I would, once again, my part of my outreach efforts is to help everyone realize that they can engage. And I would, you may be doing some fantastic seminal science and be truly on top of your field, but the people at FDA are truly on top of their field with regards to helping you turn your thoughts and your observations into a drug that's reproducibly manufactured and meets all the regulatory requirements that are out there. And there's a good reason that we have those regulatory requirements so that drugs are safe and effective. And I would engage, and that would be at every opportunity that I had. I wouldn't nickel and dime the FDA, but if we have a program, I would avail yourself to those programs, and particularly to all the. There's so much educational guidance out there that you do yourself a disservice not to step up to the plate and truly read that, and then you can ask much more intelligent questions to the FDA. Thanks for the question.
Thank you so much for that response as well. We had another question come in for Dr. Emily Warren. Could you kindly confirm whether the draft GLP tox report can be submitted at the time of initial IND for any types of INDs, so oncology, non-oncology, bio, or small molecule? Also, the final signed GLP tox report, should that be submitted 120 days after IND clearance or after IND submission?
Thank you for that question. So yes, the draft toxicology report can be submitted for first-in-human INDs for any type of product, whether it's a biologic or a small molecule. And to clarify, the final signed quality assured GLP toxicology report is expected within 120 days of when FDA receives your IND. Thank you.
Thank you so much for that response as well. And we'll go back to Dr. Patel for another question. Do FIH phase 1 studies drugs actually need to be GMP based on the phase 1 CMC guidance?
Thanks. So this question kind of touches a little bit on the first question that I answered related to CGMPs. Again, for phase 1 studies, there's a specific guidance titled CGMPs for phase 1 investigational drugs. And that outlines the requirements. So while a phase 1 study does not have to specifically, don't have to specifically follow the regulations per CFR 210 and 211, the concepts are still the same. Essentially, you have to have focus on the requirements in 3 areas: having well-defined written procedures, adequately controlled equipment and manufacturing environments, and then considering making sure there's accurate and consistent recording of data and manufacturing. Now, as you move on in clinical studies to phase 2 and phase 3, those phases of development are subject to parts 210 and 211 per the regulations. So really what you want to do in phase 1 is to assure that you are manufacturing and essentially under the concepts of CGMP. Thank you.
Thank you so much for that information you've shared. We now will address a question for Dr. Caitlin Tidings. Dr. Tidings, when is an IND exemption applicable?
Thanks for the question. So IND exemptions are something that can be quite relevant to academic sponsors as they apply to drugs that are already lawfully marketed in the United States. So the overarching concepts to understand on whether an IND can be exempt are, 1, can the trial results be used to support future marketing of the product or products by the manufacturer? And 2, is there an increased risk to the patients in the trial compared to the patients in the general population receiving the marketed drug? So if the answer is yes or maybe to either one, then an IND will not be exempt. We can help, investigators can determine whether an IND should be exempt, but if there's any question, it's always best just to submit it to FDA, and we can help determine if it will be exempt or not. Thank you.
Thank you so much for that response as well. We do have another question for you, Dr. Tidings. Could you please explain why FDA may be moving away from maximum tolerated dose approach?
Sure. So the MTD is a bit of an older concept that we used for dose escalation designs, and it was largely associated with using cytotoxic agents such as chemotherapy. And so now we have a lot of newer drugs who have different mechanisms of action other than cytotoxicity. And so collectively, we're working hard to find ways to better optimize dose selection. And several of us have mentioned Project Optimus, which is undertaking this task. So MTD may not be well served, or I'm sorry, modern anti-cancer drugs may not be well served by the MTD approach. And so we recommend working with FDA to help with that dose to optimization process.
Great. Thank you so much for that information. We may have time for one more question. This question came in for Dr. Warren. If enough publicly available non-clinical information is available for a toxic payload for an ADC, whether non-clinical, ADME, and toxicology studies with the payload for the ADC is needed for the IND filing, what kind of meeting is recommended to get more feedback on this?
Thank you for that question. Yes, so you can rely on product-specific literature to describe the toxicity of a payload for which there's clinical experience, and you can rely on that at the first in human stage, so for your initial IND filing. However, when you reach the marketing stage, you cannot rely on product-specific literature unless you have the right of reference for that data. And to get more feedback on this, you can request a Type B pre-IND meeting. Thank you.
Okay, thank you so much. We might be able to fit in one more question for Dr. Summers as well. Dr. Summers, would you recommend contacting the division office prior to submitting the formal meeting request?
Thanks for the question, Nora. Sorry to perseverate on the guidance documents, but there's a whole guidance document specifically on meeting management. And I haven't read it recently, but I'm assuming in there it tells you exactly how to go about requesting specific meetings that are formal meetings. So they would be pre-IND and above. And the Interact meeting is a reasonably new meeting, and I can't actually tell you what that mechanism is. I am assume the pre-IND is similar to the pre-IND meeting. But for the OREG stuff, if you go to the Oncology Center of Excellence's webpage, and in there is a Project Catalyst webpage, and it tells you how to address questions to us with regards to the oncology regulatory expertise and early guidance program, which is much easier and much less formal. So I would, one, read the guidance document, and two, go to the webpage. Thanks.
Great. Thank you again for that response. So it looks like that is all the questions we have time for in this current Q&A panel. Thank you again to our presenters for all the excellent information. Now we'll take a short break and resume at 12:35 Eastern Time.